Oui, bonjour.
Et bonjour à tous et merci d'être ici aujourd'hui pour le projet de thèse de Hengyi. So thank you everyone for being here for the PhD proposal of Hengyi. Aujourd'hui, donc, Hengyi va nous présenter son proposé pour une présentation d'à peu près 20 à 30 minutes où il va nous expliquer un petit peu ce qu'il planifie de faire pendant son doctorat. Et ensuite, on va ouvrir la séance aux questions pour l'audience. So then, after the presentation of Hengyi, if someone has some question, you could ask him. And then we will ask you to leave for the round of questions where we will see how Hengyi built this project and we'll ask you a question for 2 rounds. So our first round with the committee first, and then the supervision team, and then the second round if there is other question and comments. Donc, 2 tours de questions une fois que Hengyi aura fait sa présentation. Donc, Hengyi, étudiant au doctorat, supervisé par moi-même, par Miguel Montoro et par Milla Rautio, et on a la joie aujourd'hui d'accueillir dans son comité d'évaluation, M. Guillaume, qui est professeur à Umeå University en Suède, et Bernadette Pinella-Loul, professeure émérite de l'Université de Montréal. Donc, merci beaucoup d'être ici pour le projet d'Hengyi aujourd'hui. Thank you very much for being here and being part of this evaluation committee. We're very happy. So now we'll let Hengyi speak about this project. We'll just share your presentation. That everyone can see it? Perfect. Then I will let you.
So thank you very much for Guillaume's introduction. Hello, everyone. This is Hengyi, a third circle student from Biogeochemistry, directed by Dr. Guillaume Grosbois, Dr. Milla Rautio, and Dr. Miguel Montoro. Thank you very much for participation of my committee, Dr. Yang Carlson and Dr. Bernadette Pinella-Loul. Today, I will present my proposal, the impact of forest harvesting on the ecology of boreal forest lake. So let's talk about some background information. The boreal forest in Canada is the 2nd largest terrestrial biome in the world, covering an area of 270 million hectares, which is a quarter of the world's forest, as well as 11% of the Earth's terrestrial surface. Due to the importance of timber, the forest exploitation is one of the most important economic activities in Canada, and the harvesting pressure on boreal biome has been increasing in recent years significantly. So the boreal lakes are an important part in a boreal forest landscape, and there are more than 1.5 million lakes representing the largest freshwater on Earth. However, the ecosystem, aquatic and terrestrial ecosystem are usually considered as independent, and there is a misunderstanding and there is some knowledge gap in understanding their interaction. But it is important to deeply understand their interaction, as the terrestrial or allochthonous source is considered to be the major fraction of the total aquatic dump, which is dissolved organic matters in boreal lakes. In recent years, the situation is not that optimistic. Boreal forest ecosystem change have been observed due to the global climate warm changing, and also the anthropogenic activities. It has not only affect the primary production, but also has the discernible consequence for the whole tropical web and humans. So my project will focus on one of the anthropogenic activities, the forest harvesting. As you can see on the picture, the forest harvesting can increase the organic matters extracted from the terrestrial to aquatic environment. And as a result, the DOC, which is the dissolved organic carbon and nutrients, can increase by 2 to 3 times. And the forest harvesting can increase the metabolism and the organic matter process. My project will focus on the will focus on the part within the red square, and also especially on the food webs of phytoplankton, zooplankton fish, which is my chapter 3, and the microbe of bacterial to nanoflashlight, which is my chapter 2. And there is not too much study about the Mixotrophy community, and my project will also study about something about this part. So the browning is the most obvious influence from Qidong to the lake, and the Qidong, which is the terrestrial dissolved organic matters, diversely affects the lake biology. The Qidong absolutely causes the lake browning, and the lake browning absorbs light, which can largely limit primary production. On the other hand, the nutrients come with dome, and it can be promoting to a certain extent the primary production. So here's a figure. There are some studies shows the results on the top part of the curve, and our study lakes will perfectly lays on the upraising slope and down decreasing slope, which are respectively the lakes on Eskers and lakes on clays. And these two kinds of lakes are the two main kinds of lakes in Abitibi. So here, let's to get some more information about the Abitibi region and these two kinds of lakes. So the Abitibi region lays on the clay belt, which is a large tract of fertile soil stretching between Quebec and Ontario, as you can see on the map. At the same time, there are 9 main eskers, which are complex geological structure formed by glaciers during the last ice age. And the lakes on eskers and lakes on clays are the two main kinds of lakes in boreal forest of Abitibi region, and the water quality and aquatic biota will be strongly influenced by the catchment soils and the geological background. At the same time, the lakes on clays are strongly influenced by their watershed, and the lakes on clay, as they have close connection to the underground water in eskers, and their position is relatively higher, so they are partly influenced by their watershed. So we will observe the response to these two kinds of lakes to forest harvesting. So in this project, we aim to evaluate the impact of forest harvesting on the functioning of the boreal lake, and at the same time, we will study the impact of forest harvesting on lakes on eskers and lakes on clays. My project will be composed by three parts. The first chapter will be the lake biochemistry and carbon cycle. The second chapter is about the role of Mixotrophy and in boreal lakes, and chapter 3 will be the Foodweb structure and electronic. We generally hypothesize that the harvesting impact the ecology of the boreal lake, including sedimentation, greenhouse gases emission, Mixotrophic community, and Foodweb structure. We hypothesize at the same time that lakes on eskers are more sensitive and less resilient to the harvesting than lakes on clay. So here comes the connection of the three chapters. Let's imagine this is a lake. The blue part is the water body, and the dark yellow part at the bottom is the sediment. As we know that forest harvesting can increase the flux of Qidong and nutrients from watershed to water. So correspondingly, the concentration of DOC and nutrients will increase, and also the water will be browner. There are also some particles entering the water body, so we set up a sediment trap at the deepest point of the lake. This is also a part of my chapter 1, and also the particle organic carbon participates to the sediment. It will inspire the fermentation, and the methane emission will increase. The increased dome will also be beneficial to bacterial production, which will be praised by eutrophic nanoflashlight, and the mesotrophic community may have more eutrophic individuals. Bacteria and flashlight could also be the prey of zooplankton and fish. So the zooplankton and fish could contain more electronic because of the diet shift. And for the first one, the other one, the blue one, could change the phytoplankton's photosynthesis will be influenced by the sebum, and because of the dual effect of phytoplankton and also the mesotrophic community, the emission of carbon dioxide will change. So in a word, the cause or the culprit of my project is the Qidong, and the first chapter will focus on the lake biogeochemistry and also the fate of the carbon. The second chapter is about the mesotrophic community, and the third chapter is about the electronic in Foodweb. So for my project design, 12 lakes were chosen in Abitibi region with similar physiographic condition and easy to access. 6 lakes on clays and 6 lakes on Eskers. Among the 6 lakes of each type, 3 were taken as controls, which means they are located in an undisturbed forest. And for the other 3, the forest harvesting will be done from last winter to this spring. The sampling before harvesting have been finished last October, and there will be a continuous sampling for the next 2 years in order to investigate the post-forest condition of post-harvesting condition. The frequency of harvesting could be 3 times a year from spring via summer to autumn to account for seasonality. As you can see, the site of chosen lakes on the map here, and all the lakes are not far from our core city Amos, and it's about within 1 hour drive. The sampling, the fieldwork sampling will done together, and the analysis will be separated by chapters. For chapter 1, biogeochemistry. The boreal lakes can store and outgas a majority of carbon exported from carbon, and it is important role for the global carbon circle. And boreal forest can be both carbon sink for sequestering carbon to the sediment, and also the source of carbon for greenhouse gases emission. And the harvesting will not only change the biogeochemical parameters in the water, they will also bring some particles. A previous study have shown that the sediment yield will be 7 times higher in some certain freshwater body due to the harvesting. So in chapter 1, we are questioning how will forest harvesting influence the biogeochemical circle of a lake, and we aim to estimate the fate of forest carbon exported to the aquatic habitat after harvesting. And we hypothesized at chapter 1 that the forest harvesting will include DOC, nutrients, as well as the sedimentation and the production of greenhouse gases in boreal lakes. To achieve this goal and to test the hypothesis, we will take some in-situ measurement for some physical parameters like temperature, pH, and light penetration. We'll also collect the gas samples, and also we'll leave sediment trap at each lake. We'll also take back some water samples to lab for further studies like parameters like nutrients, DOC, DIC, premium production, and also algal biomass. We have a lot of geochemical parameters. So here is a table to briefly introduce their analysis. As we have talked, the physical parameters will be measured in situ by the sensors, and also the turbidity will be presented by set sheet depth, and the bathymetry will be recorded by several depth measurements. The algal biomass, which is represented by a chlorophyll, will be filtered by through a 0.7 fiber glass fiber filter. and conserving under -20 degrees until the spectral photometric analysis. And the samples of nutrients and DOC-DSC will be sent to Guillaume, analyzed by a chromatography, and DOC will also analyzed by a spectral photometry at UGAS, and CDOM, the colorful dissolved organic matters will be quantified as 320 nanometers. And SUVA, specific UV absorbance, which is an indicator of our aromatic matters, will also be an index to assess the terrestrial carbon. And we will send the greenhouse gases samples to the University of Lethbridge. So for the analysis, based on the previous study, we here predict the potential analysis that we might apply during my chapter 1. We'll do the analysis like the ANOVA test to check the difference between before and post harvesting condition, and also between lakes on clays and the lakes on Eskers. We'll do the regression analysis on environmental variables, biotic parameters, and also the carbon dioxide and sediments. And we will also analyze the underlying correlation between biogeochemical parameters and seasonality by PCA. So here comes the chapter 2, the Mixotrophy. It may explain, it may be the possible reason for the increase of carbon dioxide emission. The Mixotrophy are abundant in freshwater plankton and its main role in the food web. As you can see the pictures, you can find the trophic level of Mixotrophy. Mixotrophy mainly prey on bacterial. The success of Mixotrophy over autotrophy may vary by different environmental conditions, and it has been experimentally demonstrated that the Mixotrophy overcome of Mixotrophy to aeotroph and autotroph will under per nutrient condition, which is our boreal lakes on the Eskers. And the Mixotrophy is the main role of the metabolism of a lake, and this may be the course to turn a lake from a carbon sink to carbon source. In this chapter, we aim to assess the role of Mixotrophy in lake metabolism, and we hypothesize that the forest harvesting will change lake metabolism from autotrophy to aeotrophy due to the Mixotrophic community modification. For the Mixotrophic samples, we will collect from integrated epithelium and pass through the 53 micrometers poured and conserved with glutaraldehyde, and until the specimen preparation. For the preparation, we will use the DNA fluorescence stain in order to observe under the inverted fluorescent microscope with certain filtered light. As you can see the pictures on the left, the autotroph is formed like a coffee bean with a bright pigment. And we will also count the quantity of Mixotrophy in a certain zone to estimate their abundance, and we will also calculate their biomass by their different sizes. In this part, we will do the ANOVA test to check the difference between and before harvesting condition, and also the lakes on Esker and lakes on clay, and also we'll do a PCA to check the main characters for the changes in Mixotrophic community. For the chapter 3 food web, there are 3 main leading roles: the algae, the zooplankton, and also the fish. The CDOM will reduce the algae growth, as we have talked just now, and zooplankton is also sensitive to the environmental change, and it has been proved that harvesting could decrease zooplankton abundance and biomass. And also for the fish diet, it will be shifted because of the intervention of allophthalmi, and the contribution of organic matters to the top consumer, which is the fish biomass production increase with DOC concentration in water. So in this chapter, we aim to determine the food web structure, allophony, and production after harvesting. We hypothesize that the forest harvesting will simplify the structure of lake food web, increase aquatic organism allophony, and we have hypothesized as well upgrading of Qidong, reduce allochthonous carbon production by algae in the lake on clay, and with a contrary impact in lake on Eskers. So for these three leading roles, we will collect two kinds of samples. The first samples is for analysis of stable isotope and fatty acid, and the second samples will be done for the identification of the species and also the morphological changes. So we will call it sample 1 and sample 2 for easier. For the sample 1 of phytoplankton, we'll collect from filtered system matters, and for the sample 2 of phytoplankton, we will take the water from lake and we'll conserve with Lugo solution. And for the sample 1 of zooplankton, it will sift through 50 micrometers pores and conserve under 80 degrees. And for the taxonomy and abundance, it will be conserved with addition of 90% of ethanol. And for the fish, the stable isotope and fatty acid samples will be the fish methods will be used for the stable isotope and fatty acid analysis, and we will choose 10 fish of every species, and we will count the quantity of fish by the lakeside. And also at the same time, we will do measure their length, their bulk length, and also weigh them. For the stable isotope analysis, we will also collect the dead leaves from lakeside and also the microphytes in the lake to get the reference carbon source. And for stable isotope analysis, we will use the isotope ratio mass spectral, and fatty acids analysis was conducted by gas chromatography. For the stable isotope signature, we will use the 13 carbon to locate the production base of food web and 15 nitrogen for the tropic level. The condition analysis of carbon and nitrogen sometimes will cause some problems in analyzing their results. So we will use deuterium for addition. For fatty acid profile, we'll use the polyunsaturated fatty acid, especially the EPA and DHA, which can only be produced by the algae. For this part, we will do the morphological observation, and also we will do the taxonomy on phytoplankton, zooplankton, and fish. We'll do the same ANOVA test like the previous two chapters, and we'll do the Bayesian analysis for stable isotope to take into phytoplankton, terrestrial, and microphytes, carbon source contributing to zooplankton and fish. So my project, I hope my project will build a connection between terrestrial and aquatic ecosystem and revise the paradigm of forest management. And at the same time, I hope it will help government to make political decision making. Here is my schedule. As you can see, my fieldwork and network will last for 3 years. I have already done my before harvesting analysis. And I think that after harvesting sampling will be beginning at the end of the next month. And I will have some required courses at the first 2 years, and I will start drafting my 3 chapters, and that is from the 3rd year. At the same time, during my study, I hope to have some internship and also join an academic conference, because this can really widen my view. So I want to show my gratitude for my supervisors, Guillaume, Milla, Rautio, and Miguel, Girona. And it's a great honor to have you here, Dr. Yang Carlson and Dr. Bernadette Binet-Alou. I'd like to say thank you to my fieldwork group, and you can see the pictures here. And also, I want to show my appreciation to my fellows in Abitibi and also those from Chigudini. So thank you very much. Merci pour votre attention.
Est-ce que quelqu'un aurait des questions pour Hengyi? Mélanie.
Just a little one. On your first slide, you mentioned that the boreal forest was the second terrestrial biome. What's bigger than the boreal forest? What's the biggest one?
Oh, it's the boreal forest in Canada is the second largest terrestrial biome. The biggest one is the Russian.
Okay, you just separated them. Okay, I was wondering.
Other question for Hengyi?
Hello?
Oui?
Oui, bonjour. I would like to congratulate the student, about his very clear presentation, and he has managed to address most of the comment I gave on the report, okay? And I have now some question about the... what?
Est-ce que je peux juste, parce qu'on a besoin que ce soit à huis clos après les questions du comité. Donc il faut que je fasse sortir tout le monde d'abord.
D'accord.
Juste quelques secondes et merci. Est-ce qu'on a des questions à distance? Do we have some question remotely? No hands? Right, okay. So I will ask you to just leave the room. And...